Hello students, this is Biologia and I am Saurabh Mukherjee. Today I am talking about the main DNA replicating enzyme of prokaryotes, that is DNA polymerase III. So in this discussion I will talking about the DNA polymerase III holoenzyme as well as core enzyme. But before starting my discussion, my request to all of you to subscribe my channel and press the bell icon for quick notification. So basically DNA polymerase III enzyme enzyme resembles a hand-like structure. So here this is the 2D structure of DNA polymerase, that is 2D structure, and this is a 3D structure. This structure is a 3D structure. So in this picture, this is a template strand and this strand is the newly synthesized strand. Newly synthesized strand. Basically the core enzyme, that is the DNA polymerase III, looks like a hand-like structure where the DNA or newly synthesized DNA grips the primer or in a template junction. This slide contains the composition of the DNA Pol III holoenzyme. So holoenzyme contain the core enzyme, that is this is the core enzyme. The holoenzyme contains 3 copies of the DNA Pol III core enzyme and 1 copy of the 5 subunit sliding clamp loader. So this is a sliding clamp loader and this one is sliding clamp. So sliding clamp loader contains 5 subunit and this is a tau protein. So this is a tau protein. This tau protein is joined by the sliding clamp loader with a flexible linker. So here is the subunit of full DNA Pol III enzyme or DNA Pol III holoenzyme. Pol III holoenzyme contains 3 core enzyme, 1 linker protein. Here linker protein present in number 3, which I discussed earlier. 5 subunit clamp loader and 1 beta sliding clamp. So the core enzyme contain the 3 subunit, that is alpha, epsilon and sigma. The linker protein is the tau protein which attached to the core enzyme and the sliding clamp loader via flexible linker. The clamp loader contain 5 subunit, that is gamma, delta, delta dot, phi and x, and the sliding clamp is the beta sliding clamp which loaded in the DNA Pol III holoenzyme by sliding clamp loader. Here is the activity or here is the function of all subunits. Here a model is represent that is trombone model which coordinating the replication by DNA polymerase in E. coli. So in the first picture we see that is 3 DNA core enzymes. So this is a leading strand, leading, and this one is the lagging strand. First of all, DNA Pol III holoenzyme, that is the total enzyme interacts with the DNA helicase. So this is the DNA helicase through the tau subunit. This is the tau subunit, which also bind the DNA Pol III core enzyme. So basically how these 3 core enzymes coordinate the replication process, which I discuss here. So one strand synthesis the one core enzyme synthesis the leading strand, So leading strand synthesis is the continue process. So one DNA Pol III is engaged to synthesize the leading strand. Another DNA Pol III enzyme engaged to the synthesis of lagging strand. So another DNA Pol III enzyme engaged to synthesis the lagging strand. Synthesis the lagging strand. Periodically, DNA primase associate with the DNA helicase and synthesis a new RNA primer. On the lagging strand template. So this is the new RNA primer. Immediately after a new RNA primer is synthesized, the sliding clamp loader assembles a sliding DNA clamp at the resulting primer template junction. The unengaged second lagging strand DNA polymerase rapidly recognizes the loaded sliding clamp and initiates a newly Okazaki fragment. The first lagging strand that is synthesis another DNA core enzyme that is DNA polymerase releases from DNA and the sliding clamp after competition of an Okazaki fragment. So this cycle process is continued and DNA replication process is continued. So thank you students, all the best.